Can you imagine recording cellular electrophysiology in vitro and in vivo? Greetings, my name is Paola Morales Calvajar and I will be presenting the microfabrication and assembly of a 3D microelectrode array for simultaneous optical and electrical probing of an electrogenic organ-on-a-chip model. This research was conducted at the University of Central Florida. The cellular electrophysiology is the study of the electrical properties of biological tissues. Changes in ionic permeability of cell membranes translate into electrical activity that regulates cellular physiology. Several biological phenomena based on electrical activity are cell cardiac action, cell proliferation, neurotransmitters, and among others activities. The 3D electrophysiological recordings has a very important role in the electrophysiological field. So there has been the tissues that we record or cellular are 3 dimensions, like the hippocampus brain slide, the organoids, and the recording of a DRGS plant, and the microfluid channels. Since there are 3 dimensionals, to get the recording, it has to be penetrated or recorded inside these projections. So to get the recording, it has to be 3-dimensional. Thus, the application can be transmitted to the organ-on-a-chip models. That these are microfluidics. So we don't now in the field. The problem is that there's no the recordings that we get are 2-dimensional in nature. So the microelectrode array can be used to measure the electrical activities. Glass as a substrate material can be used to get the optical clarity. And the packaging of an MEA is needed to interface with the commercial amplification system. And for glass substance, the packaging implies a conducted via transduction. So these are complex and requires a glass DRIE and a wheel field technique PCB and or wire bonding to transmit the signals. So the microelectrode array are planar in nature. It needs to be interfaced with the recording and simulate to get the electrical activity. So this disease on a chip or organ on a chip can be used to measure the Parkinson, epilepsy, ALS, neuropathic pain, autism, and autism spectrum. So enabling the 3D functionality in the MEA will enable the extraction of optical and electrical data from the 3D cell culture. So we help realize that this is on a plate and a fully organ on a chip can be applied to the 3-dimensional microelectrode array. So the 3D microelectrode array we present, it has to be a glass on a chip for optical clarity. The 3D stereosync microelectrodes for the electrical probing, and the 3D printed frame because we want to transmit the signal with the commercial amplification system. And the device was characterized with a measure of HL1 cells, and it can be measured with other electrogenic cells. So in the methods, we first started to fabricate the 3D microneedle. So with a stainless steel sheet with IR laser, we make like a keyhole with 10 different needles. So then with a needle, we make it 3-dimensional. So since we want only the keyhole design, then we was isolated with laser again, and this is the mass that it was left with of the microelectrode arrays. So this is a SEM image of how it shows in real life the 10 different microelectrodes. So since it was made with the hand or with a needle to make 3-dimensional, the degrees we want to measure if there was the degrees were consistent of each of the other electrodes because we want to make it the 90 degrees possible. So we get a bus plot. We measure a bus plot. We have in the highest degree of 90 of 90 degrees, the variation angle, and with the lowest degree of 86 degrees and having a mean of 89 degrees of the microelectrodes. So the MEA realization, we have a glass substrate and we deposit with the E-beam on gold to make these traces. So we want in the middle of these traces, there were 10 different area. So when we paste it with the microelectrodes, they can have 10 different signals. So since the microelectrodes are conductive, it need to be conductive, we need a conductive paste to transfer these signals. So we paste it with a gold paste to be exact, and then we paste it in the middle of the arrays. But if you think about it, we paste it with a conductive paste all over the keyhole design. So we're not having 10 different electrodes. We're gonna have one electrode because they're interconnected with the conductive paste. So that is when we passed laser from every electrode to get 10 different electrodes. And of course we put a PEG ring, so when we put the cells, it can be in the same place. So this is how we show the MEA with the array with the electrodes. And this SEM image shows how it was exact the IR laser passed from every one electrode to the other. So it was a very precise method. So this shows how was the MEA after it was passed with the IR laser, having 10 different electrodes. So we want to make a control precision casting to insulate the electrodes. So we drop cast the PDMS and then we let it rest because we want only the tip to be exposed to the tissue. So this SEM image shows how the tip was exposed for the tissue and having the 10 different electrodes. So only the tissue touched only the tip of the electrode. So the packaging with a 3D printed frame, we may end the conductive via because it needs to be put in a commercial amplification system. We need to transfer these signals. So we 3D printed a frame. So when it has 10 different vias, so we want to make this vias to be conductive, and we put paste, silver paste, on every channel, so it can transfer the signal from the top to the other side where is the commercial amplification system. So this shape shows how it looks the 3D conductive of the bias. So then when we paste it with the silver paste, we wanted to adapt it to the commercial amplification system and to transfer these signals. So we want the variation of the resistance of every via, and we have a mean of approximately 4 ohms. So the 3D MEA testing was measured the impedance and phase. And in the first graph, I want to look at the difference of the package MEA, the assemblage MEA, but with the measurements we got, and the results are in literature. So there's no difference when we compare from one to another. At the same time, with the frequency, we got the frequency response, and there's no difference with the assemble MEA and the package MEA. This shows that when we adapt the 3D printed frame, there's no make difference when having the impedance and with the frequency. So in theory, this MEA shows the Randall equivalent circuit, and in the table it shows the difference with the assemble and package MEA, and there's no big difference when we got the results, because so that means with the 3D printed frame, when we adapt it, there's not gonna have difference for the results. So since we want to interface the MEA with the commercial amplification system, that is why we got this exact design. So that's when we get the 3D printed frame, because if you see, we want to the recordings we got from the top from the electrodes, we want to transfer to the commercial amplification system, and that's what the PEG ring was exposed, because we want to see the optical clarity of the cells when it interacts with the commercial amplification system. So this is the noise we got without the cells, which means that we actually having recording from the electrodes. And the 3D MEA realization has specific uses. It can be adapted to the human nerve on a chip. We made a collaboration with the same company, and they made this nerve on a chip, and they actually have the electrical recording from 2 electrodes, but it can be applied with our MEA because it can have 10 different recordings. They're gonna have 10 different recordings, so they can send one signal from one electrode and receive the signal from the other electrodes. So that makes an advance to their company, and they're recording with the human nerve on a chip. And we characterize with the HL1 cells to characterize the data of the microelectrodes array. So we got the measure of electrophysiological activity from cardiac cells, and this shows the live cardiac cells, and then this is the noise we got, the beating we got when we adapted to the MEA with the commercial amplification system. So that is the noise with 1 second with the cardiac cells. So it actually shows us that they were leaving the cells were leaving, and before research, different three-dimensional microelectrode array, the cells were actually dying. So we made an advance again to this application. So in conclusion, with the optical probing in addition to electrical probing with the 3D printed MEA, there were no different. And we have developed a technology using laser micromachining, metallization, 3D print asking, conductive bias. So for the interrogation with the organ on a chip model, and the electrophysiological data from the HL1 cells, cardiac cells, were made, and we got the cells leaving, and we got the beating from the cells. I would like to acknowledge the UCF for bringing me the opportunity to conduct this research, the RAU site for accepting me, and the members of the Nanobiosciences and System Laboratory for giving me the open doors to conduct this research. So I would like to highlight that this project was recently published at RSC Advanced at the Royal Society of Chemistry, and in subject matter has a provisional patent. These are the reference, and thank you for your time, and I will happily to take any questions.